Our today's topic is Genomics and Human Genomic Project. Question arises, what is genomic? Study of genome, it is called as genomics. Then what is genome? It is actually complete genetic information or DNA of an organism, it is called as genome, or haploid set of chromosome. It is called as genome. See here, diploid set is shown. That is, in case of human being, 2n is equal to 46. 46 chromosomes are present or 23 pairs are present. 23, it is a genome of human chromosome made up of DNA. And DNA contains gene. This analysis, sequencing and mapping of the genome, it is called as genomics. Means suppose this is one chromosome. How many nucleotides are present in this chromosome? Then which genes are located? Where they are located? This study is called as genomic. Let's see this in detail. Human Genome Project or HGP, cracking down the live code, means all genes they are studied, sequence of nitrogen bases of each gene is studied, then position of each gene is discovered, and it is used for many purposes. Genomics, the term genome, introduced by scientist H. Winkler in 1920. Actually, what is genome? It is the total genetic constitution of an organism, or it is complete copy of genetic information, that is DNA, or one complete set of chromosome, monoploid or haploid of an organism. Monoploid means one set or haploid means one set. That is, for example, in case of human being, haploid set means 23 chromosomes. The term genomics coined by T.H. Roderick in 1986. Actually, what is genomics? The study of genome through analysis, sequencing and mapping of gene along with the study of their function. See, analysis, then sequencing means which nucleotide bases are present, and then mapping, what is length of that gene, then study of their function. For example, suppose this is one gene, then what is length of that gene, which nitrogen base pair they are present, then what is function of this gene. This study, it is called as genomics, the study of genome through analysis, sequencing and mapping of gene along with the study of their function. The sequencing of yeast, Drosophila and mouse genome was done in order to facilitate comparative studies between human and other organisms commonly used for genetic studies in laboratory. You know that Drosophila melanogaster, tiny fruit fly. This fruit fly used by T.H. Morgan for the study of genetics. Then yeast Saccharomyces cerevisiae, it is unicellular fungus, and mouse, it is closest to the man. So, to compare between genome of human and all these organisms, study of genome of these all organisms, it is done. Several additional genomes are now either actively being sequenced or strongly considered for sequencing. They include several microbes, bee, tomato and other crops. This will be considered for crop improvement, then improvement of microbes or improvements of organism for benefit of human. Genomics classification. Two types, structural genomics. It involves mapping, sequencing and analysis of genome. See here, mapping Then sequencing of nucleotide. It is done in the structural genomics. And in functional genomics, function of all gene sequences and their expression in organism. For example, insulin secreting gene. Function is it stimulate or it secrete insulin, and expression, that is insulin is secreted. Function secretion of insulin, and it is actually secreted when the gene is switched on. So genomic classified into two branches, structural and functional genomics. Application of genomic. Improvement of cropland, human health and livestock. How we will improve cropland? For genetic engineering, which is the most recent branch of biotechnology, in which gene of one organism, it is inserted into other organism, or it is manipulated into other organism. Purpose: improvement of cropland, improvement of livestock like cows, buffalo, to improve their milk yielding capacity. Then human health. If we insert insulin secreting gene into the diabetic patient or offspring of the diabetic patient, at that time that healthy gene will avoid diabetes in that offspring. This is application of genomic. The knowledge and understanding acquired from genomic research can be applied in number of sectors, including medicine, biotechnology and social science. It helps in treatment of genetic disorder through gene therapy, which I already explained about the diabetic patient. Many genetic disorders are there, like sickle cell anemia, hemophilia. They are corrected through the gene therapy. Genomic is used in agriculture to develop transgenic crop having more desirable character. Transgenic means organism with foreign gene. It is called as transgenic organism. Here we obtain many transgenic crops. For example, Brassica napens. This Brassica napens, it contains gene of leech, and product, it is obtained from that seed of this Brassica napens, and that product is used to treat heart diseases. Then genetic markers developed in genomic have application in forensic analysis. Genomic can lead to introduce new gene into microbes to produce enzyme, therapeutic proteins and even biofuel. Through genetic engineering, we obtain transgenic microbes which produce desired product. We get insulin from the bacteria E. coli, which is transgenic one. So we obtain different product by this genetic engineering. But when we obtain this product, when we know the structure of gene and function of gene. Structure means their location, then their nitrogen base sequence, that is their nucleotide sequence, etc. And through genomic, these important things we get, like we get different product, we get improved crop plants, then it is also used in gene therapy for treating different genetic disorder, etc. Then what is HGP or Human Genomic Project? In Human Genome Project, human genome is studied, means structure and function of all gene of human, it is studied in this HGP. Human genome, say here sequence is shown. It is like a book. Human Genome Project. When it was started? In 1990. Who started it? International Administration of the Human Genome organization, that is HUGO, and this project was coordinated by US Department of Energy and National Institute of Health. Many countries they are involved in this project, such as United Kingdom, France, Germany, Japan, India and China. It is began in 1990 and completed in 2003. Aims of this project: mapping the entire genome at the level of nucleotide sequence. Means suppose there is chromosome number 1, it contains how many nucleotides, which genes are located on this chromosome, what is length of each gene, what is function of each gene. Then to store the information collected from the project in the database. That information obtained, it is stored. To develop tools and technique for analysis of the data. Then 4. Transfer of related technology to private sectors such as industries. For example, insulin secreting gene, then data about this gene, it is transferred to the industries for manufacture of insulin, which we need on large scale for treating diabetes. Taking care of legal, ethical and social issues which may arise from the project. HGP closely associated with rapid development of a new era in biology called as bioinformatics. In bioinformatics, all information it is collected and data it is supplied to the computer and that data is analyzed. The work of Human Genome Project has allowed researchers to begin to understand the blueprint in building and constructing the human genome. As researchers learn more about the function of genes, protein, this knowledge will have a major impact in the field like medicine, biotechnology and life science. Therefore, HGP is very important. Already we studied in medicine, that is gene therapy, in biotechnology, like a field like genetic engineering. This is very important project. Human Genome Project was to provide a complete and accurate sequence of 3 billion DNA base pair that make up the human genome and to find out the estimated number of human genes. Now about 33,000 genes have been estimated to be present in human being. How many genes are there? 33,000. And how many base pairs are there? 3 billion DNA base pairs are there, which make up the human genome. The project was also aimed to sequence the genome of several other organisms, such as bacteria E. coli, then a free-living non-pathogenic nematode, Caenorhabditis elegans, then yeast, that is Saccharomyces cerevisiae, Drosophila fruit fly, plants like rice and Arabidopsis, Mus musculus, that is mouse. Complete genome sequences of these model organism will be used for comparative study that will allow researchers to study gene function in this organism. See what scientists obtain after the study of genome of these all organisms. See here, in human, how many chromosomes are there? 46. Genes 33,000 and base pair 3 billion. You will surprised to see the chromosome number of mouse, which is very smaller than human being. In mouse, chromosome number 40 and genes 25,000. 25,000 means only 8,000 less than that of human being. And base pair 2.9 billion base pairs are there. In fruit fly, Drosophila melanogaster, chromosome number 8, genes 13,000, base pair 165 million. In plant, Arabidopsis, chromosome 10, gene 25,000. See here in mouse, chromosome 40, here chromosome only 10. But genes are similar in these both. Then estimated base pair 157 million. Roundworm, chromosome number 12, gene number 19,000 and base pair 97 million. In Is chromosome 32. It is unicellular fungus. Though it consists of single cell, it is having 32 chromosome, gene 6000 and base pair 12 million. And bacteria E. coli, only one chromosome but genes 4400 and base pair 4.6 million. And this suggests that the secret of our complexity may lie not in the number of genes but how we use them. Though we are complex, but our complexity, it doesn't lie in our chromosome number but in line how we use them. It will lead to understanding of gene structure and function in other species. Since we possess many of the genes, some of the genes same as these of flies, roundworm and mice, such study will lead to a greater understanding of human evolution. Thank you, friends.